Hello, everyone. My name is Zachary Laborde. I'm a PhD student at Indiana University. I was apparently the cause of all the bad things from the second presentation you saw. And I'm also here to present another presentation, which I hope you find good, on spatial embedding of edges in a synaptic generative model of C. elegans. So moving straight on, a little bit of background. First of all, neurons can do some really interesting things. They've been able to coordinate to form efficient global structures while just using only local information, while being able to create global efficiencies. On top of this, these connections can vary greatly between individuals. In fact, Whitley et al. In 2021 found that almost 50% of all the connections and even something as simple as C. elegans vary between individuals. Yet we have these consistencies between them as well. So with all of this in mind, we were wondering if we could create a minimal model to analyze the process of neurite growth and synaptogenesis in C. elegans. So first of all, there are two different parts to this that we're going to go over. First, we're going to go over the data, then we'll go over a model before we get into the results. Zooming in on the data, some of you might be asking, why did we choose C. elegans? Well, we chose it because it has multiple complete microconnectomes that have been reported throughout multiple different papers. So it makes it easy for analysis. Zooming in here, here is the complete microconnectome. This one's from White et al. In 1986. And if we zoom in even further, we'll look at the frontal ganglia. This is the area that we'll be looking at today. And if you notice this teal little ring here, it's known as the nerve ring. And what's special about this is that over half of all neural processes pass through this. So this makes it a very dense area as far as neural construction goes in C. elegans. Looking at one particular neuron now, there are a few different things that we have to establish about C. elegans neurons, because they are a little different from what a lot of people are familiar with traditional neurons. First of all, these neurons have 1 to 2 neurites. Now, I mentioned neurites before, but what neurites are essentially just, they act as both axons and dendrites. They're much more simple than you would expect in, a human neuron. Furthermore, these neurites rarely branch. This is known as single process. And the majority of the connections are what are known as en passant connections. Now, en passant connections are essentially just when two neurites pass by each other, they can form an electrical gap junction. That's referred to as an en passant. And then finally, in the frontal ganglia, these neurons wrap around the pharynx, which is this little green structure here in the middle that you can see. And with those 4 things in mind, we have constraints on what neurons can and can't do in C. elegans. Now, getting back out, looking at the data, we got our 3D positions of neurons from Izquierdo et al. In 2022, and our connectomes from Cook et al. 2019, as well as Whitley et al. In 2021. All right, that's it for the data. Moving on to the model. But first, we actually have to look at a past model before then. In 2010, Itzhak and Lauzun presented the Random Distance Dependent Attachment Model, or RDDAM. And why do I find this model interesting? Well, it's because it takes very minimal information about just using neuron locations, and it's able to fairly predict a lot of the network aspects of a C. elegans connectome. And for those that are wondering, the equation for that is shown right there on the bottom left. But essentially what it does is that it's exponentially more likely to form connections between neurons that are closer to it than neurons that are farther away from it. And distance is determined based off of the location of the center of their soma. So with that, they were able to find they were able to compare clustering coefficient, average distance, average connectivity, bidirectional links. All of these things we'll get further into later on, but they had some limitations. They used only 2D information, for example, and they also had difficulty predicting some of the network statistics that were shown, such as clustering coefficient. So going back into the model, here it is. We first need C. elegans again, and we're going to strip away all the complicated bits down to just the nodes. And to make it even simpler, first, it's called the Spatially Embedded Edge Model, or SEAM. And to simplify it even further, we're going to just cut it down to, let's say, 6 neurons. All right. And so here you can see the nerve ring is represented right in the middle, as we showed before. And essentially how it works is that first it will draw a ray to the nearest neuron opposite of the nerve ring, trying to replicate the proficiency of the favoritism towards going through the nerve ring. Next, it will detect the intersections between these rays, the artificial neurites. And then from there, it forms a new network that is made up of these potential connections. And then finally from here, now that we have all the potential connections, we can create sub-networks from that, given an X number of directed edges. The number of directed edges is determined based off of what we would expect to see in an actual C. elegans. And we also have a part 2, a second model. To account for the bias in neurite projection for the step 1, we also created REEM, or the randomly embedded edge model. And with this one, it only changes one step. This time, it draws a random ray. So these rays can go, these artificial neurites can go in almost any possible direction. And this is to see if the bias in choosing our selection method may have impacted the outcoming results. Okay, finally, we have results time, almost. One more thing. I mentioned this before, but we actually have to go over the network statistics, just so that everyone is familiar with everything. First, clustering coefficient is essentially how interconnected a node's neighbors are to each other. We have edge distance, which is very simple. It's just the distance between the actual 2 connected nodes. We have node connectivity. This one's the most complicated one. It's basically the number of non-repeating paths between each 2 nodes. For example, when you walk to the conference, you could have gone multiple ways here from your hotel room. And so each possible way that you went would count towards node connectivity, as long as they're not creating loops. And then finally, we have the number of bidirectional links, which is just when when 2 nodes connect to each other in both directions. Okay, finally, we're at results. For real this time. So first of all, looking at network statistical results, we first looked at clustering coefficient and found that our model, SEAM, as well as the random distance dependent attachment model were closest to C. elegans, and that basically that the proximity alone might explain the clustering patterns it was just the random distance dependent model. That is a typo. I don't know how that got in here. But essentially, if you look at the actual onto the right, you can see that there are these red lines. Those red lines are where we would expect C. elegans to be, where C. elegans was. And then the violin plots are the actual distributions based off of each individual model. So in the first one, the clustering coefficient was closest for the random distance dependent attachment model. And that this shows that proximity alone might just explain the clustering patterns in C. elegans. Next, we looked at edge distance, and here we found that both of our models were closest to C. elegans in that way, suggesting that spatial embedding neurites and SOMA alone could explain the characteristics of this network. And unsurprisingly, we found that the random distance attachment model had much more shorter edges on average, given its favoritism towards short connections. Next, we looked at the average connectivity, the thing we talked about earlier about make different possible paths. And with that, that was the most interesting in that the average connectivity was very low for C. elegans, indicating that there was low redundancy in the networks. None of them got even close to it. And this is consistent with previous work finding that C. elegans might contain a rich club, making it more metabolically efficient, essentially meaning that there is a small set of nodes that are interconnected with each other and are highly connected across the whole network. And then finally, we looked at bidirectional links. And here again, the RDDAM matched it almost perfectly. But it is unclear if this is simply a result of gap junctions, which are always bidirectional, or if this is reflected in chemical synapses as well. Next, we looked at distributions. So first we looked at the degree distributions of the networks, and what we found was that that every model except for what was in green, which is a random network, were pretty close to it. None of them were able to represent the high degree nodes that was found in C. elegans, which again could be indicative of a rich flow. Next, we also looked at the distance distributions. And from here, what we found was that our model and the random networks were close to C. elegans. This was found before with looking at previous data that RDDAM preferred very close connections. So with both of those, if we look at the closeness between both of them, the Wasserstein distances, we can see that our two models were a close balance between these two distribution methods. Next, we also decided to take a more subjective approach to it by just looking at the actual networks themselves. So we took a random sampling of each network from each model and compared it to an actual C. elegans network. And if we are to actually look in through the middle, this would be where the pharynx is. Now, obviously, these connections aren't moving directly through the pharynx, but rather wrapping around it. And in this case, the SEAM as well as the RDAM both did not well represent that middle the wrapping around of neurites around the pharynx, indicating that the C. elegans neural network isn't as simple as just looking at spatial proximity, that there is more going on. In fact, it was it looked more similar to a random network than it did to the other two. Next, we also just decided to look at the overlaps for fun. These models weren't meant to try to represent what C. elegans was doing perfectly, but rather to capture some of the aspects of C. elegans. But we decided just for fun to see how well they actually overlap. And they didn't overlap well at all. None of the models actually did a good job at actually accurately capturing all of the exact connections that were found in C. elegans. So going on to conclusions, we found that embedding neurites is an important factor in explaining certain network aspects of the C. elegans frontal ganglia. That geometry is an important factor in determining the connections in C. elegans, but it is not the only factor that matters. All of the models tested failed to effectively replicate average connectivity, which was particularly interesting because this implied that the biological neurons create networks that are much more metabolically efficient than we would typically expect in almost any network. And finally, the results of spatial embedding and comparing edge overlaps show that despite similarities of the models that C. elegans has in the frontal ganglia, they're not representative of C. elegans neural network. Again, this was accepted from the beginning that we weren't going to, accurately represent a actual real neural network, but it's still important to note. So going on to future directions, we have two different routes of ideas here. First, we're thinking of possibly doing a connection feedback approach to try to capture the what we found with the average node connectivity. Basically, how are these neurons able to know that a neuron farther away from it is connected to some of its neighbors? There has to be some sort of transmission of information in order for that to occur. And then second, another approach that we're interested in pursuing is an axon budding model approach, where because neurites form symmetrical and complex paths, can we try to predict how the axon budding occurs and put into a model? There's previous work that Randy Beer had done on it that I'm particularly interested in moving forward. And yeah, I'd like to thank the NSF as well as my institution, Indiana University, for helping funding my research as well as funding to getting me here today. I'd like to thank my PIA, Eduardo Izquierdo, as well as all these beautiful people in my lab. And the GitHub, all the code is available on my GitHub, and you can also scan that QR code if you'd like. And feel free to email me with any questions or concerns or collaborations that you're interested in pursuing. Thank you.